I'm Dr. Russell Broadus. I'm a professor of pathology at MD Anderson Cancer Center, where I've worked since 1999.
So, while you were in medical school, how often did you apply your knowledge gained from introductory biology courses?
Actually, quite a bit. Medical school, um, medical school in theory, they teach you that you can major in anything and you don't have to major in science. But the reality is it's extremely difficult if you don't have a good science background. So I did have a few friends in class who were not science majors in medical school. And while in the end they did okay, it was a big struggle for them. So you do rely on a lot of your introductory science courses in medical school.
How integral would you say that your general biology courses is like for your chosen specialty?
It's very integral. In pathology, we study mechanisms of disease, and we use the microscope a lot to get those mechanisms of disease. And I have to know what a nucleus looks like. I have to know what a cell membrane looks like. I have to know where growth factor receptors are, and you learn all of that in your introductory and general biology courses. Now, I don't pause to think, oh, I learned this back in freshman biology. I mean, it just becomes second nature to you. But all those types of things you learn in early biology, I use every day.
How do you see concepts such as like the cell cycle implicated in your work?
Cell cycle is actually very important. As an example, in breast cancer, so when we diagnose a woman with breast cancer, it's knowing that she has breast cancer isn't the only thing we need to know. We also need to know specific molecular features of her cancer because it helps to predict her prognosis and what treatment she gets. So one of the markers we look for is a nuclear protein called Ki67, and it's an indirect marker of cell cycle. The more Ki67 protein expression there is in a tumor, the more her breast cancer is turning. What I would say turning over, the faster her cells are dividing. So the faster the cells are dividing, the more aggressive, the more biologically aggressive the breast cancer is. I mean, that's just one example. That's to me, I think one of the more important examples of where we use importance of cell cycle every day in cancer biology and cancer medicine.
What kind of schooling and education did you have to go through to get
Um,
where you are now?
okay, so beyond high school, 4 years of college, I majored in microbiology and minored in chemistry. I did medical school, but in medical school, I was part of an MD PhD program because I knew I wanted to do research of human disease. And so, MD PhD program was 4 years in medical school. It was a total of 7 years, so about 3.5 years in med school and 3.5 years of graduate school. And after that, I did residency because when you graduate medical school, you get an MD, but the degree really doesn't mean anything until you do residency. So I did residency in pathology in Houston, and that was 4 years. And then that takes me to 1998, and that's when I started. That's when I became a faculty member at Baylor for a year before I moved to MD Anderson. So count 4 years of college, 7 years of medical schools, 11, 4 years of residency, 15 years of education.
Describe your favorite science class.
My favorite science class, I think, in college was actually microbial physiology. It was a senior level. The interviewer here is kind of looking like, what is this? It was a senior level microbiology class, and it was like physiology of microorganisms. And we learned all the biochemistry of these different aerobes and anaerobes, and we had to learn all these life cycles for them and all the biochemistry cycles and what substrates they grew on and how many carbon chains they needed. And it was actually a very, very interesting class. And the professor who taught the class loved students, and he was a great professor. It was a really fun very, very difficult class, but a class I liked a lot. But it was very challenging.
What led you to choose pathology?
I think ever since high school, I liked looking in a microscope. I had a advanced biology class in when I was a senior in high school, and for 6 weeks we did a microbiology part, and we had to use a microscope and look at organisms that we cultured in petri dishes. And I just thought I was a real nerd. I thought it was fascinating. I would come in at lunchtime, and after we saw the football films from that week, I would come in and eat lunch real fast, and then look in the microscope for the rest of the hour. And then also, my mom and dad used to take us to um, book fairs once a month in El Paso, and I bought, they would have really cheap books for sale. And one time I bought a pathology textbook. And this is like when I was in seventh or eighth grade. And I didn't understand anything in the book, but I liked all the gross pictures because they had pictures of all the microscopic things you were supposed to see. And so that's when I got really interested. Then I did a lot more microscope work in my courses in college, and I just loved it. And so I got hooked on pathology at a very early age, I think. But in medical school, um, I liked a lot of my rotations in medical school. I liked internal medicine. I liked psychiatry. I liked infectious disease. I liked oncology. So I was not all that sure I would go into pathology. But when when I really started thinking about what kind of residency I wanted to do after medical school, I thought about my different med school rotations and what I liked about them. And what I really found out what I liked about them was, I liked diagnosing patients, but I didn't like treating the disease they had. Once I figured out the disease, it wasn't so interesting to me. So, for when you're interested in that, being a diagnostic pathologist is probably the best, is the best career choice.
What is the focus of your research?
In our research lab, we're interested in endometrial cancer, which is a cancer of the epithelial lining of the uterus. And what we're interested in studying are what are the different molecular mechanisms for how endometrial cancer spreads and metastasizes. Because when it's localized, it can be cured by surgery and it's not such a big problem. But when it's spread outside the uterus and metastasizes, it's essentially incurable. And so, um, since I've been at MD Anderson, we've discovered a number of different molecular pathways that we think are very important for how endometrial cancer spreads.
The ultimate goal you have for your research?
I think the ultimate goal is, I think all of us at MD Anderson, our ultimate goal is, and at least my ultimate goal, is that we want to have an impact on helping patients. Am I going to cure cancer in my lifetime? No, I'm not going to cure cancer. But are we going to make a difference in treatment or in diagnosis or in helping patients? I think absolutely we will. And I think we've already done that. And that's, to me, is the best part of our job, is that when we're doing research, it's not just for gaining scientific knowledge, it's gaining scientific knowledge to apply to patients and to help them out. And that's our real goal. And I think, to me, that's the most gratifying part of it.
Basic biology play a role in your research?
Basic biology comes up every day in our research. So as an example, one of the postdoctoral fellows in my lab, Jessica, is working with a very basic enzyme that's important in the production of adenosine. So adenosine is one of the molecules that goes into forming ATP. So how often do you talk about ATP in your general biology classes? It's all the time. Or in in biochemistry, you talk about ATP all the time. So it's a very fundamental molecule in biology, and that's very important in our research right now. When we discover new genes that are important in endometrial cancer, one of the fundamental questions we ask is, where is this protein normally expressed? Is it nuclear, cytoplasmic, or membrane? And it's very important for us to figure that out, because then that helps tell us where the function is. So again, it's very, very basic biology. Is it a nuclear protein? Is it a cytoplasmic protein? Or is it a membrane protein? So, um, although we deal with very complicated and sophisticated issues every day, a lot of them can be boiled down to very, very basic biology that you have to have a firm grasp of.
What have you learned from your research?
Uh, I think we've learned several things. One of just globally, what I've learned personally over the years, and what I try to teach other students is, in research, it's very important to form a good hypothesis about your scientific problem, and that test that hypothesis in the lab. But it's extremely important to what I call don't get married to your hypothesis, and what that means is I see some students in some research just fall in love with they think their hypothesis is going to be correct, and they can test it in the lab and they're going to be proved correct. Sometimes, you form your hypothesis, you do the experiments, and you're incorrect. You have all the proper controls, you do the proper experiments, and you're proved wrong. What I always tell students, what I've really learned is you have to listen to your results, and you see what your results are telling you. And if your results are telling you to go in a new direction, form a new hypothesis, that's what you have to do. And as simple as that sounds, it's actually a very, very difficult thing sometimes. I see a lot of students struggle with that, and we've struggled with that in the past. We think we have a really good hypothesis, a good story we're trying to develop, and we do the experiments, and it's not correct. And when that happens, you have to regroup, think about the results that you've gotten, look at the literature, and reform a different hypothesis.
What are some lab techniques and tools you commonly use?
We commonly use PCR, so polymerase chain reaction, to amplify DNA. If we didn't have that, we wouldn't be able to do our work. We frequently use a different type of PCR called quantitative reverse transcriptase PCR, where we're quantifying the amount of message RNA. That allows us to get an idea of what kind of gene expression is going on in tumor cells. We use microarray analyses. That's a way to get an idea of what kind of gene expression is occurring in thousands of genes in a tumor. We use immunohistochemistry, in which we apply antibodies to sections of tissues or into cell lines. And these antibodies can be fluorescent tagged or tagged with a chromogen, so that when the antibody binds the protein, we can see where that protein is expressed in the cell. Again, if you have a good antibody, that's a really, really powerful technique. We even use an older technique sometimes called electron microscopy. It's kind of a technique that was really big in maybe the 50s or 60s, has died off a little bit, but it's still a very, very powerful technique that you can use to help localize expression of proteins to the ultrastructure of cells.
Favorite part about what you do?
I think the favorite part about what we do is there's certain lines of our research that I would say are extremely important, that if we can finish them are going to make a great impact on patients and on health of people who have cancer, or help even prevent them from developing cancer. So that's exciting. But then also sometimes realizing that you're the only person in the world working on this. And that's a really, really neat feeling sometimes, is that you feel unique, and you feel like, if I can do this, I'm the only one in the world doing this, and I can make a real impact on the health of cancer patients around the world. So to me, that's what the best part about what we do.
Was there ever a moment that you regretted your decision of going to med school or becoming a doctor?
So like when you're studying for your exams in medical school, you regret that a lot. I think though you also medical school and graduate school are funny things. They challenge you so much it begins playing tricks with your mind. The tests are extremely difficult. It's a lot of information thrown at you at the same time, and you really start to doubt whether you can do it or not. And I think most of the students in the class were all previously the smartest kid in their class in high school, or smartest kid in their class in college. Now all of a sudden there's 200 of you in this classroom, and the first test you get is like a 72, and you go, oh my gosh, this is a C. I've never gotten a C in my entire life. And then you look around and everyone else in the class got like a 70 or 76, so they're all getting the same grade. So the whole purpose is to challenge you, to make you learn more. And I think at first you really do start doubting, you start doubting yourself. But I think the important thing is to remember is that, many people before you have gone on to medical school and been able to finish it. So you just have to, kind of get the willpower to finish it. And it's all worthwhile because looking back on it, I don't regret anything. It's all very difficult to do, but it's also very worthwhile to do. And I love my job. I don't even consider it work sometimes. It's hard, but it's a treat to be able to work with students and with other clinical trainees. To me, it's a lot of fun, and I get a lot of joy out of doing that.